Welcome, and thank you for joining us today. We have families visiting from Spain, Canada, Malta, Mexico, Moldova, and across the United States. We are so thrilled to have the opportunity to meet and to strengthen the bonds within our community. With only slightly more than a year under our belts, we have made great gains in patient representation to provide the scientific community with a better understanding of our patients. This is so important. Look what we can accomplish together. I would like to thank our Parent Advisory Board, many of whom have become friends over the past year, for their support in rounding up our community, fundraising, and helping to get the word out about this project. A special thank you to the Claflin family for sharing their home and their generosity with us for a most memorable evening last night. Thank you, Caroline. We are proud to support patient advocacy and education, and so excited to welcome our esteemed guest speakers who have joined us today to shed more light on MEF2C. We would like to introduce our first guest speaker, a leader in the field, Dr. Wendy Chung. We are filled with gratitude for all of your work on behalf of the rare disease community. We are so grateful for your support. Please give a warm round of applause to Dr. Wendy Chung.
Thank you, Anne. It's really my honor to be here today, and we've enjoyed working with you over the last couple days, and just Anne, Caroline, the leadership team has done an amazing job of getting us all here, so thank you for everything that you do. For those of you who don't know, this has been powered by groups coming together. My team here at Boston Children's Hospital and the team from Simon Searchlight. We've been doing Simon Searchlight for the last 14 years, and we're going strong in terms of going forward. And you're part of a much larger community in the rare disease space for neurogenetic conditions. And the good thing is, with the power of the Simons Foundation behind us, there's a lot that we can do internationally together to move things forward. And you're going to hear today first from a couple of us about more of the day-to-day things that impact you, and then you're going to hear about some of the basic science behind MEF2C, and all of this will come together as we think about trying to plan for new treatments. And the work that we do today is critical in terms of planning for clinical trials and how we can design those in the best way possible with your input. Your input is key to be able to understand what's important to you so that we get the right things that we're solving for. We're addressing your needs in terms of doing that. Just to give you an example of the people that are behind the scenes, most of whom you haven't met, but we've got a large team of individuals on Simon Searchlight, only a fraction of whom are here today, but many of whom are back wherever they are at home, powering all of what we do. As we're doing this, the Simons Foundation, in addition, sponsors and actually gives grants to be able to support researchers who are using the data that we're talking about today. So they really think about it holistically. And what we're doing with you is to be able to gather the blood samples to power their research, gathering the data to understand what's important to you and how things stand today so that we can measure how things might be different in the future. And that's what we think about in terms of clinical trials, being able to measure those differences. As we're doing this, we're learning a lot from not just you, but from other rare disease communities that share many similar features, so that as we figure out from some of them who may be a little bit farther ahead of you, we're taking those learnings and recycling them back to you so that you don't have to go through the same painful process of learning. And doing that holistically is actually, I believe, much more efficient in doing it. So, just so you know what you're doing and why you're doing it, because many of you have gone through this and been great, but you may be wondering, so why are they torturing me with this? So I'm going to try and explain why we've been torturing you with these things. So you've been doing some things online, and we have the online process because it's hard for everyone to be able to come here to Boston. So even though it might be about 10% of you in the community that are here this week, we know there are 90% of you that aren't able to come in, and we want to make this accessible to everyone. We don't want to leave anyone behind, and so Simon Searchlight is set up internationally, and we try and support this with many different languages, and of course make the barrier minimal. As we're doing this, again, I know because I've heard it from the FDA and I've heard it through other clinical trials, that people wonder, especially with children who are developing and growing, they ask me, well, would you have seen this change just because children are growing and they're developing skills? So tell me whether or not, with whatever the intervention is, prove to me that that's doing something on top of, in addition to what we already see. So I know this because we've done this for other conditions, one of which is spinal muscular atrophy, and we're following the playbook from 20 years ago to be able to move you forward faster. And we learned from that what we needed was good baseline natural history data to know what the current state is without a treatment, without a drug, without an intervention, to compare to, and we have to have that data over time. So because children are growing, we have to have the longitudinal data. So we're collecting baseline data and we're collecting annual data. And we're using the instruments that we've heard from the FDA are things that they would accept, so that as we're doing this, we have what are going to be acceptable for comparison over time. And just to be clear, all of these data are de-identified, so your privacy is protected, but they are freely available through the Simons Foundation for researchers around the world. So that means you have less work to do. You can do it once, and you can multiply it and disseminate it to many, many people around the world, all working for you. So it's a good thing, I think, in terms of being able to help more people help you. As we're doing it, I'm not going to go through in detail, but we've got people here who can help you, or you can always email into us. But there's a dashboard online that can help you know step by step what you need to be able to do. We even have a little gift card that may come in handy for the holidays in terms of appreciating and realizing that it takes your time to be able to do this. And then we give you back this information in terms of snapshots to see what's going on. And we try and do this in a very, although it's scientific data, we try and do it in an accessible way that's easy for you to understand, so that you can see where your data are going and you can see what everyone else is seeing. It's very transparent. We also have a program that we call Research Match. So researchers around the world, if they wanted to do a study for MEF2C, they have a way of immediately getting to you, but you're still in control. You still can decide how you want to spend your time, what you want to do, what you don't want to do, but it makes sure you're not out of the loop. So if it happened to be that a researcher halfway around the world had a great new idea, you can vote with your feet and you can be able to support their research, and they can be able to connect the dots and use all of your Simon Searchlight data with the new data that they're collecting. And this is true for any researchers who might be watching this. Information about how to do that is below. We also realize that many researchers don't have connections directly to you, but they want to basically take your child and study them in a dish. They want to be able to do some screens, understand the biology of what MEF2C is doing across many different people. And so we centrally have been collecting the blood samples. I know many of you have contributed to that. And we're able to make cell representations of your child, and even, and I know it sounds really bizarre, but even make, some of the researchers make basically mini brains. So they can take even from a blood sample and make something called induced pluripotential stem cells and make those into brain cells and make representations of brain cells. They're not perfect, but they're not bad, and it's certainly easier than being able to directly do a biopsy or study someone's brain directly. In doing that, I won't go through all of the details in this, but again, the Simons Foundation has been very generous about making all of this, with their generous support, available to researchers around the world to do this, so it's a multiplier for you, and MEF2C, of course, included. Now, in addition to that, sometimes the FDA or the EMA or other regulatory agencies looking at new treatments have said, well, I don't know. I need to have some independent verification that what parents are reporting is actually the truth. And it's not that I don't believe you. I just want to be very clear on that. I actually think you know better than I do in terms of what your child is doing and how they're doing. But we'd like to be able to have independent, direct observation using standardized ways that regulatory agencies accept in terms of approval. So we've built this into the things that you were doing over the last couple days here at Boston Children's. So with doing this, there are parallels to what you've been reporting with those online measures with in-person, so we can directly see with psychologists, with physical therapists, with electrophysiologists doing the EEGs, we can directly see and verify, and that way we've got an anchor in terms of what we're doing. And the plan is to, again, do this longitudinally on an approximately annual basis. And that way we're ready so that anyone who comes up with a treatment, we're ready for that clinical trial, and we can actually inform how that's set up. In doing this, again, as we're doing this, investigators who might want to partner with us and be able to use the data to inform their studies, we're happy to do all of that, but again, de-identified, so your privacy is protected. No one has to know who you are as we're doing this. As we did this, I want to just say that getting the reports back from everyone, your kids did great over the past couple days. I know it was hard. I know many of them traveled long distances and may have been off in terms of their sleep cycle, but they did a great job. And already I can tell you we got good data in terms of almost all of the measures and almost all of the individuals. So I know it took a lot of effort to do, but I hope you feel it was worth the trip, because I do feel like we've got a really good anchor and baseline for going forward. So with this, I'm going to just briefly go on to show you some of the data. I don't have hot off the presses the data from the last 2 days, but I'm going to show you the data from the Simon Searchlight that we've been collecting so that you can take this back home, and the slides will be available and the recording available to your doctors or your therapist back at home if you want to share this. So just to give you a sense, and this is all MEF2C around the world, we've had 126 people register. The first thing we do is verify that the condition is the right condition by looking at the genetic test report, and most people have made it over the finish line. You're going to hear from Jamie a little bit more about that. After that, we ask people to then fill out some of those surveys online, and we see a little bit decrement after that, and of that, about 15 people have also given blood samples before this last week, but given blood samples before, and so that we have those samples ready for researchers, and I'll show you more about that in a second. This is, and Jamie's going to go a little bit more into this, but this is a figure to show all the different variations that we see within the gene. I put this up here mostly for researchers who might be interested in studying some of these different variations, but one of the things you'll notice is that almost everyone is unique, and for that, it's very important that everyone is represented, because there could be subtle differences between the different genetic variations, and if you're the only one with that variation and you're not represented, it's possible that in some way you won't be included in terms of the strategies going forward. Hopefully, it's all sort of approximately one size fits all, but I do encourage everyone to be included to make sure that they're represented in the research. In terms of this, just to show you, it is international. There are certainly many people in the United States, but it's meant to be and certainly represents 18 different countries from around the world, and even around the United States, representing the Midwest, the West Coast, the East Coast. We're all over the place, and that's a good thing, but that's what we want to do. In doing this, again, before this week, we had 15 people who had been provided with blood samples. And again, this is mostly for the researchers. These are in the works, but these are all induced pluripotential stem cells, or those cells that will be available to researchers to be able to study brain cells in a dish. And so in doing this, this is actually a very generous support from the Simons Foundation to be able to support this, because this is otherwise a very expensive proposition to make a library like this. Okay, so that's what researchers have available to them. This is now hopefully practical information that's helpful to you. This is the information on 43 individuals in Simon Searchlight. With time, these numbers will grow. And I'm showing you along the x-axis the age distribution. So each different number represents the age of the individual, and the bar shows us how many individuals are of that age. And not surprisingly, most individuals you see are age 5 or less, and most individuals even more than that, age 20 or less. Now that's not to say there aren't individuals over the age of 20 out there with this condition. We in fact have people that are here that'll fit that. And we know there are probably many more people who are not diagnosed with this condition who are out there, but they're not necessarily getting the genetic testing to be able to give them that diagnosis. So if you happen to know of such individuals in your community, I think it is important for us to be able to know longer term what to expect. And so I do think that's a gap in our knowledge right now, is not necessarily knowing what the decade of the 30s, 40s, 50s might look like for our kiddos. And so we are trying to work towards that in other ways I can talk about later. So I'm going to go through now the different manifestations, and the bottom line is there are no surprises. So from my point of view and knowing what you've seen yourselves, I want to reassure you, you don't have to get your blood pressure up. There's nothing that's going to be worrisome, but I think it'll reaffirm what you've been wondering or thinking you've been seeing, but to realize you're not alone. Your hunches are correct. So most of this, of what we're seeing, is what I say is above the shoulders. What I mean by that is it's mostly affecting the brain and behavior. The brain does influence other parts of the body, and including the gastrointestinal tract, and so I do think we see other manifestations of this, but it's primarily a brain condition. As we see this, there are multiple ways in which it affects the brain and behavior, and it certainly does this from early on. It may even do this even before a child is born. But in doing this, we see primarily issues that are related to what I'll call thinking or cognition, issues that result in language or communication. But I think individuals are able to understand more, that is, their receptive language or their hearing and understanding is better than their expressive language or what they're able to speak or communicate out in some way. And so think about that as you just think about how your child and how to be able to bring out the best in your child. Many of the individuals also will have what I call dual diagnoses. So in terms of their behavior, they may have other things where they have trouble focusing. So they may have an ADHD diagnosis, or they may have trouble in terms of repetitive behaviors or need for sameness or consistency. And so they may get an autism diagnosis as well. They may not, depending on where you live and what practitioners are used to being able to give in diagnoses. But it's consistent, I think, in terms of issues with understanding the world around you, processing that information, and in some cases getting anxious when you're not able to process that information. Some individuals do have other medical conditions associated with this, but many of them are secondary to the brain issues, I would say. Related to this, for instance, are issues with vision, and important to make sure that the vision issues are addressed, because if you can't accurately see the world around you, you can't process that information and integrate that information. And so important as a very fundamental thing to make sure vision is okay. In the same thing with hearing. I don't think there's a primary hearing problem, but if individuals are having ear infections and fluid building up in the ear, important to, again, process the world around them, hear language, so important to deal with those issues. Some individuals have tummy problems, so they have problems in terms of reflux, problems with diarrhea, problems with constipation. Many of those are, I think, secondary to the brain cells that go to the gut, and in terms of problems with, in the same way you notice hypotonia of the muscles, there's also can be hypotonia of the intestine as well, and can lead to some of those issues. Many of us find ways to use common medications, oftentimes that pediatricians or gastroenterologists use to treat for that. There's nothing particularly special about how we treat for that. That and dietary modifications, making sure they've got a consistent diet, and even just being active in movement can help in terms of some of the constipation. So many of those can be dealt with. Orthopedics is a common issue as well, curvature of the spine, problems in terms of foot position, needing sometimes to have orthotics, hypotonia. All of those are common orthopedic issues and very common for many other neurological conditions that we see with developmental delays or disabilities. And so I don't think there's especially sort of unique things when you're going home to see your orthopedic doctors. Many of the things they're used to seeing with children with special needs apply as well. And other problems, like I said, are seizures, and your neurologist, again, monitoring for seizures. And if you see something that you're worried about, good to bring it up with your neurologist, or even perhaps catch it with a phone, videotape it, and share it with your doctor. So being more specific with the neurological conditions, muscle tone, low muscle tone is, I would say, almost ubiquitous. Almost everyone has those types of problems. There are also problems with coordination or movement that are largely just learning to be able to move and coordinate your body. But things do come in with time. They just take a ton of repetition. And so a lot of what your therapists are doing are just repeat, repeat, repeat. And then that muscle memory eventually kicks in to be able to coordinate those activities. But be patient. Many of them will come in. It just takes time. In doing that, we also can see some other things which I'm not worried about. Some people do have a slightly larger head size. It's not hydrocephalus. It's not water on the brain. It's not something that I've seen people doing shunts for. So if you do see that the head size tends to be a little bit larger, it's okay, but I don't think it's going to need any intervention. Seizures are one thing that are important, though, for intervention. And treating those seizures and making sure your neurologist is aware about this association is important, because untreated seizures can themselves cause damage to the brain. And I don't think that this is predominantly a problem that can't be addressed. So in other words, stated another way, I think seizures can be adequately treated for most people with MEF2C, so important in terms of making them aware. But there are many different types of seizures that we're seeing, some of which can just look like your child sort of daydreaming, what we call petit mal or absence seizures. They won't necessarily jerk or convulse with their whole body, so they might be subtle. So if there is something you're concerned about, take it home to your neurologist. They can do an EEG. And I'll say that if we saw anything here that we were concerned about on the EEG, we will let you know, but it does not replace a clinical EEG. The EEG that we did here was very brief and was done for a research purpose, was not done with the intention of trying to catch seizures. So don't assume that if you don't get a call from us that you're free and clear from the seizure point of view. Do continue to check in with your doctors. The vision, as I said, the most common things were issues that are readily addressable. Sometimes people have a wandering eye or crossed eyes, and those can be treated. Some people just need glasses. They may have trouble being able to see far away. But it is important to see a good pediatric ophthalmologist, because our kids aren't able to read the eye chart and you read the letters and tell us that. And so a pediatric ophthalmologist that knows how to be able to deal with children with special needs is especially important for us. And as I was saying, there can be issues. They can be hip issues, they can be ankle or foot issues, and then as the children are going through puberty, especially spinal issues and curvature of the spine. And so when your kids are going through their growth spurt, if they haven't already, one time to have at least your pediatrician, if not an orthopedist, just check to make sure there are no problems with curvature of the spine that might need to be addressed. Oftentimes it's not that surgery is required, but there can be other things with simple bracing that can be useful. Many of the surgeries that we saw, I want to be clear, are not terrible. So a lot of things like adenoidectomies in the throat, ear tubes in the ears to be able to get the fluid out, sometimes hernias, but I'd say relatively common things for children, not in general complex things where children needed to have open heart surgery or things like cancer or major sort of things. They were relatively straightforward, so I want to reassure you about that. I put this slide up here because to me, if someone uses medication, a condition is serious enough that a parent says that I want to actually try and get some sort of be able to treat this. And I do think these issues may change over the life course. So remember that we're seeing mostly young children in terms of the report here. Seizures, not surprisingly, are something that if we see it, oftentimes we'll need medication. But some of the other things are just sleep. And sleep, I actually think, is going to be something that we can think about, and maybe even we'll be coming back to ask you how important it is and whether we need to have a quantitative way to monitor it from home. Because here, everyone's sleep is off, right? I mean, everyone's on an odd schedule. We've traveled long distances. So it would be best for us to assess sleep when you're in your own bed and you're going through your own routine. So I'm listening here. If this is something that ends up being real important for us, it may be something we come back to you, because there are ways we're thinking about with monitoring sleep from home where you could transmit the data back to us. Within this, and I say this because some people will be sharing this with their neurologist, within this, I don't think we have a home run for seizure treatment. There are many different medications that have been effective. That's the good thing. But there's one particular medication that I put here that seems to be more commonly used and more commonly effective, in case that's helpful for you to report to your doctors. Okay, very last little bit. From a developmental point of view, one of the questionnaires that you all filled out was called the Vineland Adaptive Behavioral Scale. You may or may not remember this, but you answered a lot of questions about what your child can do, and they're things that are very practical and in day-to-day life. What I'm showing you is on the x-axis, the age of the individual, and each dot is a different person, and this is what we call cross-sectional data. So everyone is represented by one data point, and it's only measured one time. But I've shown you this over the ages because you can then see what are some of the older children doing if you have a younger child, and what might you be expecting. So over time, I've broken this down. This is the first one is talking about expressive language. And as I was describing, and I put this dashed line in here so you can see where this is in terms of individuals using single words to be able to make a request. So individuals are not speaking in full sentences. So we're not seeing them, for instance, get up to the bar about telling you about an event, but there are some individuals that are developing over time skills to be able to say words. On the other hand, many individuals are able to use communication devices that even if they're not using their mouths to do this, they are communicating the same concepts through communication devices. And I will bet you that with the use of more computerized technology to be able to actually speak out of those devices, your children will be able to communicate more effectively, but we're going to need to use and work with some of the tech folks to be able to bring those to bear in terms of helping your children as we go forward. In terms of what they're doing on a day-to-day basis with taking care of themselves, they are, again, gaining skills over time. And this is now about using utensils, for instance. So rather than eating finger foods with their fingers, actually using a spoon or a fork, they are doing it. They are gaining skills over time. Practice, practice, practice. I know it's sort of really hard to be able to have the patience to do it, but practice, practice, practice really will help individuals to be able to get farther along. And then in terms of social skills, and I think this is one of the hard ones, just being able to understand what's going on and understand themselves and also understand another person, that duality is a hard thing. I think they probably are really comfortable with a small group of people in their close network, friends, family, teachers close to them, therapists, but other than that, it gets hard for them and I think sometimes unpredictable. So I think that's why we see more problems with that. With movements that are big body movements, so things like hopping, jipping, scumping, going up and down stairs, but what we call gross motor movements, individuals are able to largely gain those skills over time. We're seeing a positive slope there, but again, it is hard and it does take a lot of practice. So with this, and just the last couple slides, this one measure is broken down to young children and older children. So these are children less than 5. There are problems, I would say, ubiquitously, younger and older children about focus, being very active on the go, and with doing that, sometimes having trouble in terms of getting our kiddos to respond all the time. And so perhaps not surprising in terms of this. And again, sleep comes up here as the children are getting older. And one of the reasons I think we may want to watch this. And then again, some of these distractibility, being all over the place, being very active and going forward. So just in summary, the good news is there's not major medical problems. So what am I not seeing? I'm not seeing cancer. I'm not seeing heart disease. I'm not seeing diabetes coming up with this. I'm not seeing suddenly problems in terms of growth. So that to me is good news, that there are a lot of things we don't have to worry about. On the other hand, I'm not ignoring the fact that there are challenges, really, really significant challenges. And so what we're listening for and what we're gathering the data on are to understand what's a meaningful difference to you. And so it may not be that everything's perfect. You know, it's not like we go from here to all of a sudden jumping up here, but what is meaningful to you so that as we think about things that are sensitive to measure those changes, that's what we're going to focus in on, is getting measures that give us data that are accurate to what you're seeing, reflective, quantitative, and in real time so that we can see those changes over time. Okay, very good. Let me end there, and I'm going to we're going to run some questions. Okay, and we're going to have questions.
Hi, everyone. I'm Caroline Claflin. I'm a member of the Parent Advisory Board for the USMEF2C Foundation. Thank you, Dr. Chung, for that presentation. We have time for just a few questions. Does anyone have anything they'd like to share or ask? Yes.
Before you had gotten to the part about sleep, I had written it down to ask about it, and I just want to, it's not a question, just a statement that in the Facebook group, I've been a member for 10 years already, and that sleep difficulties comes up very frequently, so I would like to maybe emphasize that I hope you do look into it to see the correlation. Thank you.
So I'm not surprised, and thank you for the endorsement. One of the reasons, I'll tell you my justification also in terms of thinking about sleep is when during sleep, a normal sleep cycle, and with REM sleep is where you're consolidating a lot of your memories and what you're learning during the day. And so if sleep is disrupted, it actually impacts the child themselves in terms of their learning, but it also disrupts the family. And so I think that disrupting of the family is a secondary thing that then if you're not getting sleep, your day is off and your mood is off and all sorts of other things. So, and it's, I do think that if we do that, it may catalyze and sort of have a waterfall downstream effect of other things for the child, for the other members of the household. So that's part of the reason that I think sleep is also important. So thank you for telling and sharing.
How many patients participated this week in the assessments?
I think we had 31, 33, I apologize. So.
Yeah, I'll be right over.
I'd also like to know more about like feeding issues that the kids have, because I noticed there were some differences in my son and some of the kids last night. So I don't know if there was any talk about researching that a little bit more too.
Yeah. So tell me what you mean about feeding issues.
Like self-feeding, and like my son was on purees until he was about 5, and then he started with people food, but he doesn't really chew. He like swishes and swallows.
Yeah.
So he eats a variety of things, but not crunchy, and just very particular about eating, and he's not really interested in it that much either. But I noticed last night, like some of the kids were interested, so I just found that kind of curious, because he's 18, so it's been a struggle his whole time.
Yeah.
But I didn't know how many other families were impacted by struggles with the feeding piece.
Yeah. So one thing I'll say is I anticipate there are going to be cohort effects is the way I'm going to describe it. So what I mean by that is there may be generational differences, in part based on, for young children, what they had as supports when they were little. And I think it's evolving and continues to evolve, hopefully mostly in a positive direction. So depending on what people did, there may be sensory aversion in terms of having things in the mouth. And so with that, there may have just been sort of adaptation to behaviors and to things where then you sort of get used to something and you don't want to change. And I think I'm true of the same thing. So it's not just our kids, but so within this, and then it's a question of you pick your battles, like how important is this? And if it's not that important, like what's the big deal? As long as they're getting the nourishment and nutrition they need.
Yes, coming to you.
Hi, just this last couple of days I've noticed a lot of our kids have some sort of sensory processing issues, either extremely sensory seeking a lot and maybe some sensory avoidance, but it's not kind of, maybe it's lumped under autism, but I don't see any mention of it in the Simon stuff.
Yeah. I didn't call it out specifically, but you're right that it gets lumped into some of these other measures. It's absolutely the case in terms, and you see it yourselves. I think within this, it's, to me, part of the complexity about understanding and integrating the world around you, and in some cases, it just becomes overwhelming, and in terms of overstimulation, and with that, you just need to essentially run away. You need to shut it down because it's not predictable. So I think that's a lot of what's going on, and not unique only to MEF2C.
Is there another one back here?
I have two questions. So I understand right now data is relatively limited, but do you have plans to parse things out by individual variations within MEF2C? And then do you have an idea of what the actual mechanism that might be that's disrupting their sleep is?
So two things. Number one, I hope that the entire scientific community that's interested in MEF2C is going to, as I said, many people will use the data and start parsing this out by different genetic variants and if there might be the same or different mechanisms. I'm not going to presume I know the answer to that yet. I do know from we call it a power point of view, we're going to need more numbers, I think, than what we have now and more information. So I hope that this will be sort of a call to action in terms of more people participating and having enough data that the researchers can answer your question, because I think it's a critical question. So the next thing in terms of sleep, I do think we need and I'm not a sleep physiologist, there are people that think about everything from sleep initiation to sleep maintenance and different stages of sleep. And I think a first stage is to be able to understand what part of that sleep cycle is disrupted and whether it's all parts or certain portions of this. But it's actually a common feature I find to many other neurodevelopmental disorders. And within this, on the other hand, it's not universal. I think that's one of the things that's interesting to me, in trying to understand from parents what they may have done to the sleep environment, to sleep training, to other things that'll help either normalize that or help kids get through it. And what I mean by that is simply get through the night and get enough sleep.
We have time for just a couple more.
You mentioned about getting scientists or researchers like interested in MF2C. What makes one rare disease more interesting to scientists than other? And what do we need to do as a community to be the most interesting?
I'm going to let some of the scientists answer that because you're going to hear from some of them this morning. So some of them, just based on their own work, are studying, if not MEF2C, conditions that are, or genes rather, that are related to that. And so either because it's the same type of gene, it's in the same pathway, it's affecting the part of the brain, the part of development, they may be interested from that point of view. I think depending on how aggressive you want to be, then it's finding all of those researchers and raising their awareness proactively. Don't passively wait for them to come to you. You go to them and let them know about things. I've seen some family groups be very aggressive about doing that. You all can decide how you want to sort of spread the word in terms of that. I personally, the part that I'm doing is to partner with you to what I think of as de-risking it, making it easier for them to work with you to do this because they can do this and it's free, it's available, they don't have to do a lot of the hard work, we've done that with them. And then the other part of this is raising their awareness to grants that are available to be able to get funding to do this work because they need to pay their people to do this. Some foundations have funding themselves to do it. Some foundations may not have funding themselves, but they're really good at aggregating all the funding sources that are available and making those researchers aware of those opportunities, the grant deadlines, and partnering with them to be able to get in a strong application. So a couple different ways.
Here we go.
What drives you to study this gene? In your personal perception, why did you start researching MF2C?
Yep. So I'll be clear that for me, it always starts with a patient and ends with a patient. So literally, that's always the way my journey as a doctor goes. And I'll say it started with Liam in terms of thinking about this. We had already in Simon's Searchlight included MEF2C in terms of one of these about 200 conditions that we're studying. And these are based on known association with these genes with autism and other neurodevelopmental disorders. So I started this, as I was saying, about 14 years ago at the Simons Foundation, but really supercharging this really came with Liam and being able to work with you all. So.
Okay. Wonderful. Thank you so much, Dr. Chung.
Hello. I'm Kelly Olson, and I am a member of the USMEF2C Foundation Parent Advisory Board. And I'm excited to introduce Jamie Lundy, a certified genetic counselor and research coordinator from the Autism and Developmental Medicine Institute at Geisinger, to discuss genetic concepts, types of genetic variants in MEF2C, and how to make sense of a genetic lab report.
Great. Well, thank you so much for having me today. I know that I've had the opportunity over the last few days to talk with a lot of you, and I'm really happy to see a lot of your faces in the room. So I will get started with the Genetics 101 talk. Just as a presentation overview of what we'll be discussing, we'll begin with an introduction to chromosomes, DNA, and genes. We'll also talk about types of genetic variants as well as types of genetic tests. And we have a sample lab report here to break down the components of a lab report to hopefully help you all in making sense of your own lab report. And then we'll end with some other common terms to know. Sorry, I'm a little taller than the other speakers. So just from a background standpoint, starting with some background, all of our bodies are made up of millions of different cells. And if you look within any one of those cells and you break that cell open, you can find our chromosomes. Our chromosomes are known as the larger structures that help to organize all of our complex genes and our genetic information. And if you were to unwind one of those chromosomes, you could look at that classic double helix DNA structure. And the DNA provides each gene, which you can think of as an individual instruction for making a specific protein to then go out into our body to serve some specific function for us. This is a nice overview of what the chromosomes look like. So we all have 23 pairs of chromosomes. So one of each chromosome usually comes from that person's mom, whereas the other chromosome usually comes from that person's dad. And our chromosomes are numbered from longest to shortest. So you'll see the longest pair is the first pair, and the shortest pair is the 22nd pair, so those little pink guys. We all also have sex chromosomes that will determine biological sex. Typically, females have 2 X chromosomes, whereas males have 1 X chromosome and 1 little Y chromosome. And this can be important later on, and we'll break down an individual chromosome a little bit more specific soon. So if you zoom into the genes along the chromosomes, our genes are made up of what's called nucleotides, or can be thought of as letters. The possible nucleotides are A, T, G, and C. And the nucleotide code is read in what's called the triplet code, or these sets of 3. And this is important because each individual triplet is going to correspond to a very specific amino acid. Then those amino acids are strung together, much like beads on a string, to be able to be folded up into a larger, more complex protein. So putting all of this together, our DNA is going to provide the instructions for building a set of amino acids, and then those amino acids are going to be strung together to form more complex proteins. And then finally, those proteins are going to go out into our body to serve some very specific role, such as making a piece of our heart to pump blood throughout our body, or a piece of our brain that's really critical for learning and memory. So when we're thinking about our genes, of course there can be variants or changes in the way that the genes are spelt. People may have heard about the term mutations. We tend to not use mutations so much in terms of people because that can be a little bit offensive, but I tend to use genetic variants. But regardless, variants are just referring to underlying changes in the way that the DNA code is spelled. Now, there's a couple different types of genetic variants, so we'll walk through these here. The first type is what's known as copy number variants, or CNVs. Many people will use the analogy of an encyclopedia to represent our genes. And if we think about an encyclopedia as having 23 volumes representing the 23 sets of chromosomes, a copy number variant is going to be describing a deletion or a duplication of genes. So this may look something like in volume 12, chapter 13, page 302, then paragraph 2 and 3 is deleted, so representing a deletion of genetic material. We name copy number variants based on the specific chromosome location. So recall how we looked at that nice pretty picture of the chromosome spread. This here is an example of chromosome set number 5, which is where the MEF2C gene is, just for your interest. And within the chromosomes, there's 2 arms. There's a shorter arm that's called the P arm. In this figure, it's on the top. And then the longer arm of the 2 is called the Q arm. And then along each chromosomes, like rungs on a ladder, are bands. And those bands are numbered. So we can get very specific about knowing exactly where a deletion or duplication is occurring. And just for an example, how we would describe a deletion of the MEF2C gene, this would be something like a 5q14.3 deletion, because again, we're describing the chromosome, the arm, as well as the band. Whereas for a single nucleotide variant, and these are spelling changes within the genetic code. So to go back to that encyclopedia analogy, this would look something like in volume 3, chapter 15, page 300, then there should be the spelling MOM, spelling mom, but rather that last M is substituted out for a P, so now the sentence reads MOP, or MOP. So all of the content of the genes is there, but it's just a very specific misspelling in the way that the gene is spelled. Within single nucleotide variants, there's a few different types of variants, so we'll walk through a couple of these here. The first example that I have is what's called a missense variant, and this is describing a specific letter or nucleotide that's substituted out for another. Now, remember how we talked about the importance of that triplet code and those 3 sets corresponding to a specific amino acid. If we substitute one nucleotide for another, that is going to cause a different amino acid to be incorporated into the protein than was originally intended. And again, this is what's called a missense variant. Next is a nonsense variant, which is similar to a missense variant in that it is a specific letter or nucleotide that's substituted for another, but rather the effect of this substitution here is that there is a stop in how the gene is created. So there's a very specific triplet code that indicates to our bodies to stop producing the gene, and the nonsense variant means that that nucleotide substitution is going to cause that stop to happen earlier than is typical. So this would typically cause the gene to lose its function depending where exactly that stop is incorporated. Next is examples of insertions, so duplicating letters, or deletions, so deleting letters. So again, all goes back to the importance of that triplet code. So regardless of whether we are deleting letters where they shouldn't be deleted or inserting letters where they should not be inserted, the effect is going to be a frame shift variant because everything after that deletion or that insertion is going to be changed in terms of how that triplet code is read exactly. Okay, so now we'll move on to types of genetic testing. As many people may know, there unfortunately is not one perfect genetic test that can look at our genes in all the different ways. Rather, there's different types of genetic testing technologies that sometimes families have to go through to get answers. Starting from the highest level test is what's called a karyotype. So a karyotype is literally looking at the overall structures of the chromosomes to see if there's any extra chromosomes, any extra arms, but really big pieces. Whereas a microarray is similar to a karyotype, so again, looking for deletions or duplications, but it's much more specific. So it's able to actually look along those bands of the chromosomes to find much smaller deletions or duplications. Another type of genetic testing technology is sequencing genetic testing. So sequencing is reading through the individual letters or nucleotides of genes to look for any of those single nucleotide variants or very specific spelling changes. Within sequencing-based genetic testing, there's multiple types of tests, and this is just to highlight a few, but the first example is what's called a gene panel. And a gene panel will look at a group of genes related to a specific concern. So perhaps a seizure panel, a hearing loss panel, even a cancer panel for increased risk for cancer. But it's only looking at a very specific group of genes. Whereas for exome sequencing will look through all of the coding regions of all of someone's genes. So it's a much more comprehensive genetic test than a more specific gene panel typically. Okay, so now we will walk through a sample lab report to break down all of the pieces. This is a sample lab report from Ambry Genetics. I'll point out that there is no patient identifying information. This is purely a sample report, so nothing specific here. And of course, each individual genetic testing lab is going to be unique in how they look, what their style is, but they all will generally include the same pieces of information. Starting at the top, usually there will be patient demographic information, so the patient's first and last name, their date of birth, their biological sex, as well as the sample type that was tested. Perhaps they provided a cheek swab or a blood sample or even a skin biopsy for this testing. Along the top of the report, it's typically pretty clear what test was done so that we're oriented to, was this a panel, a microarray, exome sequencing, and some labs will have branded names of their tests. So Exome Next is just Ambry Genetics' branded name for exome sequencing. The other important piece that I'll point out to take note of is the date. As we're probably all aware, genetics is learning a lot very quickly, so it's important to recognize what date the result is from, just so that we're oriented to what we may or may not know. Moving down the report, typically labs will include an overall summary or interpretation of the test, whether that be positive, negative, or uncertain, where we can have a very high-level overview as far as what the test results were. Continuing to move down, and this is specific for exome sequencing, but exome sequencing requires a patient's summary. So what are their presenting symptoms, features? because since it's looking across all of the genes, it needs to be a little bit directed in terms of which genes it's giving most attention to. So it's very typical to see some indication for testing, patient summary, with the patient's features, so things like seizures, developmental differences, autism, etc., as a summary for what the test was exactly investigating. And then next, that little box is really the meat of testing with most of the information, of course, that we want. And we'll walk through these piece by piece. So starting on the left side, this example is nice in that it includes a single nucleotide variant, so that specific spelling change, as well as a copy number variant, so a deletion or a duplication. So on the top, we're oriented to that the first finding involves the EBF3 gene, and the second finding is including 2 genes, so presumably a deletion or a duplication of those genes. Next, this lab report specifically will call out if that gene has been characterized or if it has not been characterized. Again, just going back to the fact that genetics is learning a lot, sometimes there's genes found or variants found in genes that there may be very limited information about. Next is, what is the relevant associated syndrome or disorder for this specific gene? So in the first example, they're calling this EBF3-related neurodevelopmental disorder. And then in the second example for the copy number variant, again, since we name these by the chromosome location, this is called a 9q34.3 copy number gain, so a duplication of genetic material at that position. Next is the mode of inheritance, and this is for the specific gene that was identified. So what is typical for this condition? To break this down, first, autosomal dominant inheritance means that a variant is on only one copy of the gene, and if that harmful variant is present, then that is enough to cause symptoms of that related disorder or syndrome. So, of course, the MEF2C gene, we would say, follows autosomal dominant inheritance typically. You only need one harmful variant to cause symptoms. This is in contrast to autosomal recessive, which some people may have heard of in other contexts, but autosomal recessive occurs if a variant is on both copies of that gene, then that condition is said to be autosomal recessive. So things like cystic fibrosis, spinal muscular atrophy, many other conditions. So moving right along here, next is the genotype. So this breaks down some information important for that specific genetic variant. We'll start with the zygosity. The zygosity can either be heterozygous or can be homozygous. So a variant, and this refers to the specific variant that's identified, a variant is heterozygous if at that same position on the other chromosome, there's a different variant. So there's only one variant for that individual. Whereas a variant is said to be homozygous if at that same genetic position, so that other same chromosome, then there's the same variant. So we very carefully have to interpret the known inheritance pattern with the zygosity of the variant identified to see if that would cause an impact or not. Along the same vein, variants can be inherited, so they can be something that a parent has and then passes down to a child, or a variant can occur de novo or of new, and this would be a spontaneously occurring variant that just happened very early on in that child's conception. Okay, so next is the alteration or the specific genetic variant for that individual. For a single nucleotide variant, how we'll break this down is the C dot is first describing the change at the coding level. So on the right side, there's MEF2C, and then C dot is going to orient you to the nucleotide position, so what number nucleotide is affected, followed by the reference amino acid, which is the reference nucleotide, which is the nucleotide or the letter that is typically present, and then that little greater than sign is going to point to the variant nucleotide or the new nucleotide that's incorporated. So how we would describe this would be MEF2C, C.113, T to C. And also for single nucleotide variants, we describe it on the protein level. So what does that coding level impact the protein level? So this is very similar how we read with the P dot. First is the reference amino acid, or the amino acid that is typically at that position, followed by the amino acid position, what number amino acid are we talking about, and then followed by the variant amino acid, or the new amino acid that's now incorporated. So for this example, we would read it MEF2C, P dot leucine, 38 proline. And if you recall back to earlier on, then this is representing a missense variant. Okay, whereas copy number variants are going to be named by the specific breakpoints on that chromosome. So we previously walked through how we name chromosomes and how we get oriented to the specific band. So this would be 5q14.3, and then in parentheses are the larger genomic coordinates, or where exactly on that chromosome does this breakpoint occur. And then finally, how many numbers of that region in parentheses are there? So times 1 would be describing a deletion, so that region is deleted. Times 2 would be typical, so we wouldn't normally see this on a lab report. And then finally, times 3 would be a duplication, so that region is duplicated. Okay, moving right along. And then lastly will be the variant classification, or the alteration classification. So taking all information into account, what is the laboratory's understanding of the change that they identified? There's a few different variant types. So we'll start with benign, which is harmless. So this is when a genetic variant is not known to cause any health or developmental differences. And typically benign variants are not reported on lab reports because we all have probably thousands of them. Next is the pathogenic category. This means that there is enough evidence to believe that a specific variant or spelling change would be harmful and therefore causing an individual's symptoms or features. And then the gray zone category, so not quite benign but not quite pathogenic, is what's called a variant of uncertain significance. I think of these as variants that there's just not enough evidence to say either way, so they have this uncertain classification. There may be more information that we can collect, such as biochemical testing, parental testing, that can help to resolve what these mean, but we generally walk people through, or a clinician can walk people through if there's any additional follow-up that can be done for these. Okay, and then continuing to move down the lab report is usually an interpretation section, so just a brief sentence or two of the overall interpretation of the test results. Were they diagnostic? Did they provide an answer for this person, or is there additional follow-up testing that could be done? Next is a notes section. The notes section is very important because perhaps if this were research testing that needs clinical confirmation, that would be included. Genetic counseling is usually recommended to help people make sense of results, and that type of information would be found here. Okay, and then we will wrap it up here with other terms to know. So you may encounter genotype versus phenotype. We all like to throw around these terms. Genotype refers to the specific genetic variant, so what is that exact spelling change? Whereas phenotype is referring to the physical presentation of that genotype, so things like seizures, autism, etc. Mosaicism is a concept in which a genetic variant may be in some of an individual's cells, but not all of them. So sometimes a child may be described as mosaic if only some of their cells have a specific genetic variant, whereas others do not. A parallel concept to mosaicism is what's called germline mosaicism. And germline mosaicism is usually said in reference to parents. So a parent is said to be germline mosaic if a genetic variant is isolated to their sex or gamete cells, so sperm or egg. So they themselves do not have the genetic condition, but they may have multiple children that do. And I included this plug in the end, so how genetic counselors like myself can help support your families. We, of course, and as I hope that I have done today, can help to navigate complex genetic information just to foster understanding. We certainly realize that genetics can be like alphabet soup at some times, so hopefully I was a little bit helpful today. We can help in terms of walking through your individual lab report, again, just for understanding purposes, hopefully connecting similar families with each other, as community is such an important aspect of your journeys. And then at Simon Searchlight, we're always working to develop new resources, and those can be largely informed by your needs. So we'd love to hear back from you. Okay, and I'll wrap it up there. Our email is there, so if there's any follow-up questions that we don't get to, then we're happy to take them there. But thanks for your time.
Thank you so much, Jamie. Does anyone have any questions for Jamie? Yeah.
Thank you. What might it mean if the interpretation section says next generation sequencing for the sample did not pass quality thresholds required to detect gross deletions or duplications?
That is a great question. I will be happy to look at that individually, but I will say in general, when genetic testing is done, the findings usually have to be confirmed. So usually lab reports will go through a second step to confirm that what they found is true rather than an artifact or something, just a technological noise. So perhaps if they didn't have enough sample to confirm what they found, maybe. But yeah, that would be my guess. But yeah.
If somebody was, I'll just use my case as an example, we were de novo for my daughter Taylor. How might you test, you mentioned something about the germline, how might you test somebody's eggs or sperm to see if we don't carry the gene, but I guess our eggs do?
Yeah, that's a great question. So germline mosaicism can be presumed if parents are negative, yet they have multiple affected children. Of course, that's a much more like that happens after the fact versus wanting to plan for the future. So sometimes parents want that extra level of reassurance and may consider going through IVF to actually harvest eggs externally, test those eggs in a laboratory, and then implant ones that did not inherit the genetic condition. So people may want to explore that.
Would MEF2C show up in the embryo testing prior to IVF?
I would think that a lab would be able to analyze for that, but it'll be unique to the lab. Go ahead.
So if people really, so number one, this doesn't happen very often, this mosaicism that Jamie talked about, so I don't want you to get overly concerned about it. There are now ways of being able to test not the eggs, but testing the sperm. So you take a semen analysis from the gentleman, and to be able to do sequencing to see if there might be a low level of mosaicism. And if you were to find that, then some of the families will go on to do the in vitro fertilization to make that diagnosis on the embryos. Otherwise, I'd say most families end up going with a natural pregnancy and then testing during the pregnancy to see whether it's available. And there are even some labs that can do that from a maternal sample rather than having to do an amniocentesis or a chorionophil sample. But if anyone's really wants to go into that, like, catch me and we can talk about it more.
Any other questions? Great. Well, thank you so much, Jamie.
Thank you.
All right, hello everyone. I'm Tushar Putran. I'm a member of USMEF2C Foundation, Parent Advisory Board, and I'm thrilled to introduce to you Dr. Michael Greenberg, esteemed neurobiologist at Harvard Medical School. He's going to discuss with us what we can learn from MEF2C iPSC-derived neurons. Please welcome Dr. Michael Greenberg.
Now, I realize that I'm in a difficult position, and so are you, because you just heard Genetics 101, and I suspect you only heard a little, understood some of it. And now, what I was planning on doing was giving you a neuroscience lecture, which is an advanced course. We're probably at the 500 level, and I don't think that's going to be very useful. So as I was listening to the last talk from Jamie, I was thinking about what I could do and that would be useful to you. When I'm not a scientist, a neuroscientist, one of the things I do is I play the clarinet. And I play in the Brookline Community Band. And the way I do that is there's music in front of me, and I practice and practice and practice. And then I go to the sessions where we get together as a band, and I sit next to 35-year-olds, and I can't quite keep up. Now science is like that too. You have to practice and practice, so I practiced for this talk. Now, there's another way of playing the clarinet, and that is you could be a jazz musician, which means you improvise all the time and you don't really plan it. I guess you're just very good at it. So in this talk, what I decided to do is the jazz musician version, to go away from the script and to try to tell you some things that I think might be useful. And at the risk of being very jazzy, maybe a little disorganized, maybe atonal, you might not understand all of it, but I'll do my best. And so one of the things I was thinking might be useful is if you would feel free to interrupt me as we go along and just ask questions. The questions about the science would be more useful than a lot of the data I was planning on showing you, because it was really pretty high level. I was thinking I was going to give a science presentation. So, all right. I think one important point to consider here is that, well, it's well known that our sensory experience in early life, input to your brain, in the brain leads to the release of neurotransmitters that are critical for the maturation of neural circuits that underlie our learning, memory, and behavior. So there's got to be pathways where the neurotransmitters cause changes in the neurons of the brain, which then allow you to execute those functions. And I think with MEF2C, it may be that there are problems with those pathways. So that's why learning can be difficult. So now this is interesting to think about, because if MEF2C is a disorder of what I would refer to as experience-dependent learning, if these pathways are not working quite well because of the absence of MEF2C, well, that then will give you a window of vulnerability in early life, but also potentially a therapeutic opportunity, because if the signal's not getting through, it may, in a sense, be a brain in waiting for the signal. And if you can then repair that, maybe you can get the signal through, and then you can essentially mature the brain. So what's happening in the brain in early life? Well, one thing you probably heard about is, first, that at birth, the brain is immature, and in humans, further development of the brain is going to ensue over a period of decades. You know, you might hear that boys, their brains are not developed until they're well into their 30s, and that's why executive function can be. With women, it's a lot earlier, but still, it's a process that occurs over several decades. And one example of this is that many of the connections that are formed by a year of age, those connections in the brain, you might think you want to keep them all, but they're actually whittled away by a process called sensory-dependent synaptic pruning. And this happens over several decades. So you have a certain number of synapses, and then some go away. And it's not just that they are just withering. There's a genetic program. The genes and the input from the environment determine that process. And we're quite interested in how that happens.
What was the name of the process? I'm sorry.
Pardon?
What was the name of the process you just described?
The process is called synaptic pruning. And it's also referred to as synapse elimination. Synapses being the connections in the brain. Some of them are eliminated, and the ones that are used are strengthened. And you could imagine that if you knew MEF2C was important for that process of synaptic pruning, which it probably is, well, then when it's not present and can't work, it doesn't happen. So again, the brain is in an immature state. Now, one also piece of good news in a sense is that a process like this, if it's not working so well, it may be that with more input or repeated learning, you could actually overcome some of the defects. So what my laboratory has studied for many, many years now is how your sensory experiences through gene expression shape the development and maturation of the brain. And the idea here is that when the signal comes from the outside environment, it could be auditory, it could be visual, it could be through touch, that a signal is sent in the brain, which then goes to the genome and turns on a group of genes that are important for maturation of the brain. And I'm going to show you in a schematic what I mean by this. So, by the way, you can model this in a dish. If you were to take, you hear about iPS-derived neurons, these neurons in a dish. If we treat them with a stimulus that acts like a neurotransmitter, and then we ask, what happens to gene expression in those cells? We see a whole set of genes get turned on. And turned on, it means the DNA makes messenger RNA, which then makes proteins, which are the workhorses in the cell, which allow for the changes in the cell and the connections in the brain. So in response to neurotransmitters, and in a dish also, in our brains or in a dish, we see that a signal gets sent, and we have essentially a set of genes that get turned on. And I'm just going to refer to a few of these. Can you see this pointer now? Yeah. So I'm showing you a wave. This happens within several minutes after you stimulate the cell. And I should say this happens in your brain when you wake up in the morning. And it happens in the visual cortex, the region that mediates vision. And when you're learning, this happens in your brain in response to learning. And experiments we're doing in the lab with mice, if you take a mouse and you put it into a new environment, novelty, like doing the crossword puzzle or going to a new place for us, these events happen in our brains. These genes get turned on. And so you see it here. This represents the turning on over time. And I show you a bunch of genes. They have funny names, FOS, JUN, NR4A1, EGRs. We're studying all of these. And what these genes are they're encoding proteins, the workhorses, in the cell that then are going to turn on a second wave of gene expression. And the cardinal gene here is a gene called FOS, which I've spent my adult life studying. Been studying this for 40 years. You might probably, if you're tuning out right now, you wonder why would this guy study this for 40 years. So, are there questions so far? Yes.
You mentioned it's like a circuit being cut, so that signal can't get to the other side.
Yeah.
Does the amount of time that circuit hasn't gone through affect any type of treatment in the future?
I think it's a good question, and I don't think we really know the answer to that, but it's possible that it would, but it's also possible that there's still a lot of room for intervention. So let's say the normal process of learning in early life, these pathways get activated and changes in the brain ensue, right? Now, if that doesn't happen, it just may be the brain's immature, and if you were to bring something back, you could start it going. But it could be when processes don't happen, there's a certain, a lot of connectivity and things degenerate and so on. So probably the answer to your question is probably a bit of both, if I had to guess. So if you have a process that's not working, the earlier you intervene, probably the more likely it is to be able to do something. And I should say you're sitting next to Sarah Trowbridge, and who works with me and is a pediatric neurologist, so she could probably answer that question better. So another question. Okay. Oh, yes, in the back.
What would the help of an MRI to have a scan of the brain?
Yeah.
And see if it's physically all there.
Yeah.
But the signals are not sent because you have these types of difficulties.
Yeah.
Can we somehow excite the system to make it work?
Yeah. Well, so I think that's sort of the point of, a lot of the ways of treating disorders like this are by repetitive exposure and enhanced exposure and learning. And Wendy could talk more about that, but my understanding is that can be helpful, and probably one way of understanding this is that these pathways I'm talking about, if they're not working quite as well, it may be that repeated stimulation might be helpful. You could say, well, how could that be? Because if the protein's missing, the signal won't get through. There's more than, there's what's referred to as redundancy in these pathways, where if something's missing, there are other components that can compensate for it. They may not work as well, but maybe with repeated intervention, you could see some hope. Now, I hope I'm not saying anything. I'm a strict, what I would tell my mother is I'm just a PhD. I'm not an MD, and she was always dissatisfied with that fact. But so I can't really speak to the medical aspects of this other than, sort of in a very, maybe even a glib way. So others here can say more about that. Other questions? So what you're seeing here, this F stands for FOS, and that's a protein which is a transcription factor that turns on other genes, and it interacts with the J, which is called JUN, for whatever reasons. I won't go into the reasons for those names. I was thinking, by the way, that what is a challenge for folks in the audience who took high school biology and didn't like it and then didn't listen are going to have a lot of trouble paying attention to what I have to say. But I think that people would even conclude that biology is hard. It's not hard. It's a vocabulary. So it's really a sense of learning the vocabulary. If I were teaching high school biology, I would insist that you all take it 4 years in a row, so that it's like learning French. If you don't know the vocabulary, it's hard to follow this. So if this sort of thing really does seem like too much, I really would recommend getting a basic textbook and just reading it, because it'll make a big difference. Because it's vocabulary. It's not that these are hard concepts. So I don't know if that's helpful. Anyway, FOS and JUN get together when they're induced. So sensory experience in your brain, in a lot of different ways in the different regions of the brain, are going to turn on FOS and JUN, and they're going to get together, and they move into the nucleus of the cell, and they bind to a sequence on the DNA, which is shown here. It's TCA, a C or a G here, and a T, and then a TCA. And when that sequence is present, there's a capacity for these factors to bind, and then what they do is turn on a different program of gene expression. And what I'm showing you here, we call these the late response genes, because they come on, first the early genes come on, the transcription factors then turn on a second wave, and these genes encode proteins that are effector proteins. They're involved in neuronal connectivity. They're involved, for example, in that program of pruning where you get rid of synapses. So input from the brain turns on this program, and you have a lot of proteins that are here that are involved in connectivity, growing the synapses and so on. And actually, I show you some of the things that this program can regulate. One aspect of this is the maturation of the excitatory synapses in the brain that allow for these, various behavioral outcomes. And it's referred to as spine maturation. These are little protrusions on your neurons, which actually are the sites where excitatory synapses form. And this program I'm talking about, which is regulated by USMEF2C, is important for spine maturation. Another process which I mentioned is synaptic pruning. We've already gone over that, whereby initially those excitatory synapses form and then they're whittled away and eliminated. That is also an activity-dependent process. Neurotransmitters have to be released, they send the signals in the brain, and then they turn on the genes, and then these processes happen. Another one, which is very important, is recruitment of an inhibition to balance the excitatory synapses. And when you hear about epilepsy, there's too much excitation. It could be because there are problems with inhibition. And the programs I'm talking about are important for controlling inhibition. Now, perhaps the simplest thing to think about for today is that these mechanisms I'm talking about are important for plasticity, for learning, memory, and behavior. And if you block the pathway, it's not going to happen normally. So, in early studies, what we found is that when there's enough synaptic activation, when enough transmitter is released in your brain in response to your experience, a signal sent, and here's the signal. You see that? That's an action potential. And it sends a signal to the nucleus of the cell. Well, actually, what happens when the action potential is generated, the cell it gets depolarized, and these channels open up, and calcium comes into the cell, and a signal sent to the nucleus, and it turns on some genes. And I want to go over here because I want to point out. So after the transmitters are released, you've got this channel opens and calcium comes in. There you go. Calcium comes in. And this intricate pathway gets activated. And you don't want to know. This is why I say you have to study biology to really learn the language. All these different proteins get activated. A signal gets sent to the nucleus. And all of a sudden, MEF2C, which is sitting on the promoter of the FOS gene, it gets activated, and you turn on the whole program. So MEF2C sits at the center of this program. So if it's not here, the signal's not getting through, or not getting through as well. So if we really want to understand what's going on in the MEF2C deletion syndromes, we need to understand these pathways better. And it's difficult because, well, it turns out that not just MEF2C, but it turns out that if you perturb any one of a number of these proteins in the pathway, you get developmental disorders. So I would say understanding this pathway overall is going to be important because a variety of these genes give rise to developmental disorders. MEF2C being really sitting at the center of this. And yes, in the back.
So kind of very layman's term question. So is there an understanding that kind of the neurons and the synapses are like physically developing properly, and there are the synapses exist and the dendrites and the number of synaptic connections exist, but because MEF2C is missing, then
It's a maturation process, exactly right. So I was clearer than I thought. So yeah, you could think about this as the synapses form, at least initially, those at birth. And with a lot of these syndromes, things can seem normal at birth. And then when your experiences are impinging on the brain to turn on programs like this, these intricate programs, that's when things go awry. But that also makes you think that there could be reasons to think that intervention could be helpful. So I would say in this field where people are looking at these different syndromes, and there are more that fit into the pathway, because the pathway, I'm just showing you how FOS gets activated, but then FOS has to work to turn on the other program, and it has still another complex of proteins that are important there that also can be disrupted in various developmental disorders. So when you think about therapeutics, you could think about it two ways. One is, let's just bring MEF2C back through gene therapy and fix things. And that's a really good thing to be trying to do. And there's a lot of activity in the general field with gene therapy to work on each of these disorders, in at least one would hope. But the other idea, which is not a better idea, but the only one that I can be helpful with, is to say, well, if it's broken, if you're going to fix it, you have to understand it. So what we're trying to do is to understand everything we can about these pathways. And I've been at it for 40 years, and we're making progress. But one of the things that if you want to think about, let's say there are 20 different types of neurons in the brain. That's an underestimate, but general. There are different types of excitatory neurons, different types of inhibitory neurons. This program is different in each type of neuron. It's not the same. So to go back a second, this part, the part where MEF2C is involved, that's pretty much the same. But when these guys get activated, this later program is going to be different in each type of neuron, so it makes things more difficult. So, some more questions? Yes.
So, to summarize, if I'm understanding you correctly, MEF2C is like basically a bottleneck, and that signaling cascade comes down and then it hits the MEF2C gene,
Yeah.
because it's not functioning correctly, it just stops.
Yeah. And that is correct, and it's not quite stopped, but it's impaired.
Right.
You know.
Okay.
Yeah. So, and I think the thing also to consider here is that you see, it's not at the very beginning. I mean, the L-type channel at the top of it leads to something called Timothy syndrome. So that's going to affect the whole pathway. But with MEF2C, the early parts get in, but this is pretty central to this pathway, and so the rest of it doesn't occur normally, and that's something we need to understand and overcome. Now, it's going to be important to figure out what's happening there when the pathway is blocked. So, as I mentioned, MEF2C induces FOS and JUN, and they turn on a whole program, and we've been very interested in understanding that. And I don't know what else to tell you about this, but that could make some sense. Well, so FOS is a transcription factor. This is the jazz mic as opposed to the classical one. In terms of a transcription factor turns on genes, and the genes are the workhorses. So if you want to understand what FOS is doing in any given cell, you have to find out where it binds and then what genes it regulates. What genes get turned on? You know, you have 20,000 genes in the genome. Which ones are chosen for this neuron to turn on to proteins that are the workhorses? So we have ways now of figuring out where FOS binds in the genome. And it used to be complicated, but now there are methods for saying, okay, if I have a group of neurons or I take a brain from a mouse, can I fish out FOS bound to the DNA and say, where does it bind? Because where it binds is going to help you understand what it's doing. So here's the problem. There are, in your genome, about 500,000 different places where FOS could bind. And in any type of cell, about 10,000 of those are used. So it's a big question for scientists. How does it choose and what is it doing? But we can figure out where it binds. And here's an example of what it looks like. These peaks. This is a piece of the DNA along here, and we can take our cell, we can cross-link FOS to the DNA, and then we can, so it sticks where it is. We can now lyse the cell and what's called sonicate the DNA into bite-sized pieces and take an antibody to the FOS, pull it down, and then sequence the sequences, and we can see where is it bound. So that's helpful because knowing where it's bound, and we could do that for MEF2C also. You could find where in the genome is it normally binding. So one way you could get a problem is by mutate. I'm sorry, I'm a mutant. You guys are variants. Anyway, so what a variant could do is it could somehow knock out the function of MEF2C, but a variant in the DNA could also affect the binding of MEF2C in the DNA. So knowing where it binds and where in some disorders it doesn't bind can be useful. Any more questions? Yeah.
Yeah, just one question. Obviously, if I look at what you were talking about and your work, super complex, highly database oriented,
Yeah.
what could be now the impact of artificial intelligence to kind of like, what you need maybe the past 20 years to get to it? Could that be sized down to like 2 years? I mean,
Yeah,
everybody's
so-
talking about it will be huge.
Yeah.
Developments going forward. Is that helpful for your work?
Very, very much helpful. And it's a problem when you get to be my age, having to learn new things like that. But artificial intelligence and machine learning is very helpful for understanding where these factors bind and what they recognize and why they do and so on. So we're definitely using that in the lab. And there's, and I'm even trying to learn it, but I'm a little slower than I used to be. So, wind it up? Perfect, okay.
We'll take questions.
Questions, I'm done, yeah. I only got to show you 5 of the 50 slides I was planning on.
Hi, I'm Josephine from Malta. I don't know if I'm going to ask the right question, but my hope is to bring on board a question that our neurologist had with regards to hypotonia. His concerns are like it's the first time that he's working with 2 MEF2Cs, let's say. So there's no other case back home. So his question is, what I'm trying to ask. With regards to hypotonia, it's a question of chemicals like synapses, blah, blah, or it's a question of related to bones, like if it's related, if it's bony, let's say, the problem.
Yeah. So hypotonia meaning with respect to muscle, and or could it be bones? So one of the things to keep in mind about MEF2C is it's expressed in cells other than the brain. And if you know, I should have thought to say this. If you want to think about how the brain's working, you might think about how the muscles work. That if you exercise, muscles grow and you know, your abilities improve. And part of that is because the motor neurons connect to the muscle, and you're getting nerve impulses, and you're essentially strengthening your muscle. The brain is somewhat like that. Neurotransmitters on the muscle get released. Now, muscle is actually where MEF2C was first identified. So there's MEF2C in the muscle, and it could be a lot of what I'm talking about here is relevant to some of the hypotonia in the muscle, would seem to me. Now, in addition, you ask about bone. In our studies, we identified, we were looking for, you know, so I said we had this program that's gets activated. We have been studying this in the mouse for 30 years, and we decided, well, why don't we study it in human neurons also? So we got some human neurons in a dish, we stimulated them, and then we used the sequence approach to find out all the genes that get activated. And we learned about this other gene. It's called osteocrine because it's expressed in bone. And we'd never seen it in the mouse, and it appears to be induced selectively in primates. So it's a monkey human gene that gets activated in the brain. It's also activated in the muscle. It's also activated in the bone. So it could be, oh, what I didn't tell you about osteocrine is the reason it's turned on in primates and not in mice is because there's, through evolution, a new site that controls the gene which binds MEF2C. So you might wonder, so if you study MEF2C in the mouse like we do, you're completely missing this major gene in primates that's the target of MEF2C. So it'd be a good argument for working with iPSCs that human iPSCs that can be made into neurons, because there are going to be targets of MEF2C that are the effectors that allow it to function that are special to human neurons. And so, in terms of your question, it could be bone, it could be muscle, or brain, or probably to some extent. I'd put my money on the muscle and the neurons.
Yes, Tushar.
Hello. My question is, I think we have addressed MEF2C not getting produced enough, versus I want to know if we have any study wherein it's more than needed produced and it's causing too much. But I want to continue with that question on, like, another question is, we have collected so much data on this kid from an external point of view, like, this functioning is not working. And did we ever go reverse, thinking that, okay, this gene is responsible for this activity and these kids don't have that, and somehow connected back it to MEF2C that it also affects this other?
Yeah, so with a lot of these developmental disorders, you can have a problem if you have a variant that leads to a loss of function or a variant that causes duplication. And my understanding is that there are examples of duplication from F2C as well, and those need to be studied. That leads me to say something else, and that is, I talked about MEF2C in terms of a specific pathway, how experience through MEF2C turns on this program that's intricate that then affects all these synaptic changes. There are other things that MEF2C does as well. So it's complicated, and we need more research to really figure it out. Yeah, but duplications can be a problem. So when you're thinking about gene therapy, you want to be thinking about the various ways of reintroducing it that might actually be oriented towards getting the level right.
Correct.
But at the same time, we need to, if you don't understand it when it's broken, how do you fix it? We need to understand the various functions, because different individuals with variants may be different, because in one sense, MEF2C might repress genes, which it does before you ever stimulate. Others it could activate. It could interact with different cofactors in the cell. So knowing exactly what those mutations are doing will help us to know what the best therapeutic would be.
Thank you.
Time for one more question.
If the patient with MF2C do train out, do exercises and also to gain more muscle weight, mass, do they gain muscle or do they degenerate?
I would have to leave it to the physicians to answer that question. Wendy?
As far as I've seen, this is not a neurodegenerative condition, so I haven't seen anyone actually regress and lose skills over time. Again, with the limitation that most of our data are for people up to 20 years old, but I have not yet seen neurodegeneration, which is in contrast to other conditions where we've looked and we have seen that.
So if we push the patient to progress himself, so in a way we can do it.
I think it's a matter of part of its muscle mass.
By time we don't, they don't degenerate, they improve.
I think they improve, and I think part of it is muscle mass, and I think part of it is muscle memory, is what I'm going to call it, which is more in terms of the brain connection to the muscle, and that repetition and the practice is what comes in with doing it. I think in part the way I think of things is we're trying to optimize day-to-day function as much as we can, and also prepare for that, keeping that window open so that if treatments become available in the future, our kiddos are as strong and as ready as they can be if there are opportunities, whether they're technical opportunities, whether they're sort of medicine opportunities, but we're trying to keep them as strong as they can for day-to-day life as well as for opportunities in the future.
And the other question, is there osteoporosis?
Osteoporosis is the question. So in my opinion, we really haven't, that usually doesn't kick in for children. It usually kicks in for older people. And what I'm waiting to see is the more sedentary you are, so in other words, the less active you are, the more chance there is for osteoporosis. So yet it's another reason to keep moving and to keep weight bearing, so standing up and moving like that. But the honest answer is I don't know what people look like for the most part in their 50s and 60s when we see that.
You know, your question makes me think of one more thing that should be mentioned, and that is, in humans, genes like MEF2C are often parts of a family of related genes. There's a MEF2A, there's a MEF2B, there's a MEF2C, and a MEF2D. So in our neurons, A, C, and D are all functioning. So if you have no MEF2C, or in general what it is you have two copies of MEF2C, so you have less MEF2C, there are also still A and D present. And they can compensate in many of your cells. You might think, if you thought about it, what I was talking about is this central pathway. If MEF2C isn't there or is present in half the amount, you would think, oh, it's just not going to work at all. But there's A and D in many of your neurons. There are certain neurons that really rely more on MEF2C, and those are ones that one would have to look at. And I think muscle does rely more on MEF2C than some of the others. But the others are there, so there's this compensatory possibilities that one could think about. Now, when you have a family like A, C, and D, it's also important to keep in mind that the proteins are not identical. They have some differences, and understanding what those differences are could be helpful as well.
Thank you so much. We have one very last question from Stephanie, and then we'll have to move on.
Sorry. Since all of our kids have different deletions, mutations, frame shifts, duplications, there's so many different things. Unless they're completely missing the full gene, no one is the same. Is there truly a way to actually give like a one-size-fits-all to fix this, since it impacts so many genes downstream that we don't even know? Or do we have to literally look at each individual case to see what genes are impacted and how can we fix that?
Yeah, so when people in the lab ask me, should I do this experiment or should I do that? You don't have to know very much to say, well, you should do both of those experiments. And I think the answer to your question is that the different variants are going to have different effects. But in many cases, but not all, in many of the cases, it's due to what's referred to as a loss of function. MEF2C isn't working. So you could imagine if you repair it through gene therapy, there would be, in a sense, one size fits all. But not all, but a lot. But I still think that when you're thinking about a problem like this, you want to think about you want to have a backup plan. And I think a backup plan is to understand the protein and what those different variants do. A lot of times when you have a variant in different regions of the molecule, what it does is it causes the protein not to be made. Even though it's doing in different places, it can still be that the protein isn't made, and if you could bring it back or repair the mutation. So I think people are very excited about gene therapy because, in a sense, it's a one-size-fits-all, at least for MEF2C. And even beyond that, if it can work for MEF2C, similar approaches might be used for many of these other disorders. So I think that is getting a lot of attention now. And people like me who want to do the basic science, we have to fight for some of the oxygen.
My apologies, but our last speaker was originally going to be Michelle Campbell of the Neuroscience Office of the FDA, but she had a last-minute personal conflict, and we are lucky enough to have with us today Dr. Haitham Amal, Associate Professor at the School of Pharmacology and the Faculty of Medicine at the Hebrew University of Jerusalem, and you're now currently a visiting professor, is that right, here at Harvard Medical School and Boston Children's Hospital. Dr. Amal will discuss his MEF2C research with Dr. Stuart Lipton on organoids, stem cells, and mouse models, and drug development efforts stimulated by this research. Thank you.
So now I can say good afternoon, everyone. So I'm gonna talk a little bit about the efforts that I am doing with Dr. Stuart Lipton at the Scripps, specifically on MEF2C models. And later on, I'm gonna tell you a little bit about the efforts to develop a drug for autism and also diagnostic methods, blood diagnostic test for autism. And I'm gonna talk about how MEF2C is related to these efforts to develop a drug. So these are my disclaimers, and I wanna first thank, first of all, Dr. Lipton for a great collaboration. I wanna thank the Eagles Autism Foundation that funded my lab in the last 2 years, specifically working on MEF2C and SHANK3 mutation. And previously, a month ago, we got a very large grant from the California Institute for Regenerative Medicine, around the $17 million, to investigate air pollution and nitric oxide in air pollution and how it affects MEF2C and other mutations on the spectrum. So I'm going to also talk about a little bit about air pollution and environmental factors. And just talking generally, the MEF2C is, as Dr. Greenberg said, it's a transcription factor, modulates neuronal function, synaptic processes, and it is associated with other neurodevelopmental disorders. And Dr. Chung also mentioned many of the comorbidities together with MEF2C. It can be associated with ADHD, epilepsy, and other clinical symptoms. And I'm not going to go deep into it. I'm going to go directly to the science that we are doing on MEF2C. And so this collaboration is that Dr. Lipton is generating the brain organoids, as just mentioned by Dr. Chang. So we are generating a MEF2C organoids from kids that has MEF2C mutation. He sends it to me, to my lab, and we are using a very unique tool that I developed at MIT when I was a postdoc at 2015, that we can identify the modification of nitric oxide on the proteins. So we take the organoids, we process them, and we can identify what are the proteins that are modified by nitric oxide. I'm going to talk a little bit later on nitric oxide. But what we found in these organoids, on MEF2C organoids, many biological processes that are affected by this alteration of nitric oxide. And most of these processes are actually, as you can see here, metabolic biological processes, which can highlight our future vision of what are the next step of experiments that should be done in order to identify drug targets for MEF2C. And the same with pathway analysis. So there are many pathway analysis. I'm not going to go into it, but I mean, what we are trying to do is, through the big data, and AI was mentioned here, we use a lot of AI and machine learning in order to identify drug targets and biomarkers, not only for MEF2C, but for SHANK3, Casper2, Neurologen3, many different mutations. In parallel to the big data and proteomics work that we are doing, this work was done by Professor Lipton, that he shows that iPSCs generate more astrocytes and fewer neurons, and a lot of electrophysiological work that was done. And now I'm gonna switch my talk to the nitric oxide hypothesis that may shed light into the drug development efforts that we are doing. So you're seeing here to the right Professor Steven Tenenbaum. He was my mentor at MIT, and he is the one with Michael Marletta from Berkeley that discovered that nitric oxide actually is produced in our cells, in human cells. And as you can see here, they wrote an article saying serendipity and discovery from nitric oxide to Viagra. And the reason they wrote this is that when they discovered nitric oxide in the late 70s, they didn't know that nitric oxide will lead a breakthrough not only on the developing Viagra, but I mean most importantly, developing a treatment for heart diseases, hypertension, and other diseases. But also, I think they didn't know that this discovery of nitric oxide may lead to, hopefully, a treatment not only for autism, but also for other neurological disorder that I'm going to mention. So this was the first paper that was published with Dr. Goping Feng and Steven Tenenbaum when I was at MIT. So we developed this tool, and Dr. Greenberg mentioned the calcium. And as you can see here, our hypothesis is that genetic mutations leads to influx of calcium that activates the neuronal nitric oxide synthase enzyme that actually lead to a massive production of nitric oxide, which may lead to the pathology of autism. But I mean, this work that was published 5 years ago didn't do any drug development related work. And this was the follow-up paper that was published at my lab at the Hebrew University, which entitled "The No Answer for Autism Spectrum Disorder." It was published in Advanced Science, and here we actually developed a small molecule. And what we can see here, and I'm going to talk how it's related to MEF2C in a minute, that in a normal state, there's a normal production of nitric oxide, that this is actually nitric oxide is a very important molecule in the brain and in the body. But when there's an autism-like mutation, and we already know that MEF2C mutations leads to a massive increase of nitric oxide, and this is how it is linked to nitric oxide, and this is not me that discovered this, but Dr. Lipton has showed that there's a massive increase of nitric oxide, and we are seeing now in our lab that when we look for specific nitric oxide-related changes, we see these changes. And the third window to the right, you can see that when we inhibit the enzyme, we see reversal, not only on the molecular and synaptic level, but also on the behavioral level. So you would ask how you examine behavioral in mice. And for example, this is the first test that we do. The first day we put the mice and we introduce him to two identical objects. And then the second day, we replace one of the objects into novel object. And then we see whether he has a novelty-seeking behavior. And we can see that when we inhibit nitric oxide, we see reversal in this improvement of novelty-seeking. We look for sociability. And also with mice, you can introduce the mice to a stranger mice and see whether the mice is more social and wants to play with the other mice. And this is kind of indication of sociability. And also we look for anxiety-like behavior. In the bottom, you put the mice in a cross maze, and you see whether he goes to the closed arm versus the opened arm. And how it all links to different mutations on the spectrum and on MEF2C. And so our hypothesis is that since we see a similar behavioral deficits in terms of sociability, anxiety-like behavior, repetitive behavior, etc., so the hypothesis is that different mutations may lead to a similar molecular alteration, and part of them is the nitric oxide. So based on this, actually, I have co-founded, based on the results in the lab, and this is, it's linking both talks previously, that how you link the findings in the lab and translate it into a product or into industry. And we have co-founded this Irish-American company named Neuronos that the main goal is to actually develop small molecule for nitric oxide in order to reverse autism-like phenotype. And now we are doing all of the IND-enabling studies for the FDA, including toxicological studies, and hopefully within 1 or 2 years we'll start first in human. And another part of this is another company that I have established is Point 6, which actually looking for nitric oxide modification in the blood. And we are trying, using AI models to diagnose autism through blood testing, and I'm not going to go into it very deeply. But lastly, before I finish, I want to talk about air pollution and environmental factors, and this is something that debatable, partly debatable, but I mean, I'm not debating it. I think that air pollution and environmental factors lead to neurological disorders. So to the right, this is a paper that we published 2 years ago, and we gave mice to drink arsenic, and arsenic is a metal that we can find it in water and rice and baby-based products, rice, and also we can find it in water in many places, even in the United States, such as New Hampshire and Nevada. There are many epidemiological studies showing elevated arsenic in water. And what we found here, that we gave arsenic low and high doses to mice to drink for long-term and short-term, and we found biochemical and behavioral changes that are linked to neurodevelopmental disorders. And to the left, I'm going to mention the serum that in the coming 3 years, together with Dr. Lipton, we're going to look for air pollution. And the way we're going to look at this, we're going to mimic the air pollution into organoids and iPSCs and mouse models and see whether these pollution or nitric oxide and other elements in the air can lead to alteration in the brain. So I'll open it to question, but before that, I want to thank my group at the Hebrew University, that without them, nothing would happen, and all of the funding agencies that funded my work in the last 5 years. Thank you so much.
Thank you, Dr. Hummel. Does anyone have questions? One question I have, if it's okay. I'm sorry.
I don't know if I understand it correct, but behavioral issues or ADHD, it's because excessive of nitrous oxide.
This is what we have.
In our system, in the body.
Yeah. So this is the question, or?
That's a question.
Yeah. So this is what we confirmed actually with these results, that when we inhibit nitric oxide, we can see a reversal in the autism-like behavior in mice. We still didn't confirm it, of course, in human, but I mean, at least in lab, on the behavioral level, in mouse models that have human-based mutations, we see a reversal when we reduce the nitric oxide in the brain.
So, if you breathe oxygen-rich air.
Excuse me, again?
If we breathe oxygen-rich air, do you see a behavioral difference?
So I'm not expert in this field, but I hear a lot of work that is being done on hyperbaric, right? Exposure of oxygen to, yeah, and according to the papers, we see some improvement, but I'm not sure if a high exposure of oxygen may not lead to oxidative stress and to the opposite side. So I can't tell you. I'm not expert in this field, so I'm not telling you whether it's.
I wouldn't say about hyperbaric chamber, just breathing rich oxygen with a simple muscle.
So if you are so the question is, again, we can come back to Dr. Chang and see whether they see any hypo-oxygenating in the brain, and whether.
In brief, don't do the hyperbaric chambers.
Yeah, I didn't want to say that, but.
Atmospheric oxygen is perfectly fine.
I have a question, which is, with the development of these drugs, what are the symptoms of MEF2C disorders that you think could be alleviated through these sort of drugs?
Yeah, so this is a great question. So we still didn't get into the level of looking for into the mouse models of MEF2C that Dr. Lipton is, if this is what you're asking. So what we have looked meantime is only the core symptoms of autism and not any other related behavior.
Yeah, I guess I mean the core symptoms of autism. It seemed like you were inferring that sociability
Right.
and
Anxiety, memory,
anxiety,
novelty
all of those
seeking,
things
yeah.
showed promising reversals in
So
the mouse
yeah,
test.
so I assume, according to the hypothesis and according to the molecular and cellular findings that we are finding already in MEF2C organoids, I have a strong belief that when we will do when we test the molecule in the mouse model of MEF2C, we'll see some reversal, at least in these behavioral tests that we have conducted.
I have a quick question. Just curious, when the drug develops, how do you intend to, like the humans consume this, like an oral pill, or like it's going to be through?
Great question. So now we are working with LabCorp. Now we are working with LabCorp into developing the first generation. Hopefully, would be sub-Q injection of subcutaneous twice a week. And the second generation that is already under development is an oral. We hope to have an oral formulation to have it as a syrup or something similar. But I mean, all of the preclinical studies right now and also the development of the formulation with LabCorp is sub-Q injections.
Just always been curious with the medications being developed. Are there a one and done scenario, or is it going to be that you're seeing right now something that would be a continuation permanently? Like, do these mice, revert and have normal capabilities and then not have to take anything anymore? Would be my.
So according to our experiments, this would be a chronic administration of the drug, and honestly speaking, we still didn't check whether if you give, for example, a month and then leave the mice without any treatment, what will happen. But I mean, according to what we did right now, is we injected 2 months and we looked for behavioral. So this show efficacy, and in the toxic level also we don't see any toxic. So this would be the next step in the company to inject this molecule into rats and dogs to test, to examine the toxicity of this molecule.
Couple more.
Hi, you mentioned possible human trials within a couple of years. Will MEF2C kids be involved in those trials or no?
Yeah, so it's very early to decide, but I mean the phase 1 would be according to what we are planning would be on healthy adults, and then phase 2 that would be in several years. Would be planned very soon, so I'm not sure about it. Yeah.
Okay. Any other questions? So before you go, Dr. Elmwell, I actually wanted to ask all of our experts here what they think, as professionals in the field, what you think the most exciting intervention is in this space or might be in the next 10 years. I think as families we hear a lot and synthesize a lot, but I'm very curious to hear what you think will help us the most in the next decade. I won't hold you to it, I promise.
Okay. So I think there could be 3 things, and they're going to come with time. So I alluded to some of them. So number 1 is I do think there's something about unlocking our kids' ability using technologies. And so it's not medication, it's not anything else, but it's using other things to be able to help get through the ideas they have, but the communication limitation that they've got. So I'd say that's number 1. Number two is I do think there are going to be things to help with sleep. I don't think they're going to be MEF2C specific necessarily, but I do wonder if there are going to be things more downstream in terms of what may help with sleep, and to the extent that help with sleep comes with other things, I think that may be something that'll come in the shorter term as other things are coming up. The thing that I look towards, but I truly have no idea if it's going to work. So realize that, because I don't know this therapeutic window and how much time we have and how much we can reverse or help development go forward, is some of the new gene editing methods will actually not actually worry about mechanism, about whether it's loss of function or gain of function or alteration of function. It will literally erase and correct the genetic variant, and those are coming along nicely. But they suffer from the ability to get into the right cell type and to be able to get distributed uniformly throughout the brain. So there are some delivery issues that have to be solved to go forward. But if that technology becomes feasible and safe to do, and it can be done across the life course, then it is potentially a kind of one-size-fits-all when it's ready to go. I don't think MEF2C is going to be first of the brain conditions to go forward with that technology. In a good way, many other genetic conditions are looking towards the same, and so if those problems get solved in a generic sort of way, I think this community wants to be ready to jump on the bandwagon for whenever those problems are solved, so that they can be applied and get in line to be sort of at the front of the line when that becomes more generalizable. So those are the predictions. We'll see what comes to pass in the next 10 years.
Are you referring to CRISPR technology then?
It's a little complicated. I'm glad to do a little primer afterwards. I actually wouldn't do CRISPR per se. There are some technological issues with CRISPR, but I do think there are some other editing technologies that are even better.
Wonderful. Anything else that our other experts would like to add? Thank you so much. Thanks for everyone for attending. We're going to have a group photo out on the terrace as lunch begins to commence. You want to say one last thing? Sure.
Hello, I'm Jesus from the Spanish Association. I want to thank USMEF2C to organize this meeting, of course, to the experts and doctors. And I want to say that we have brought a gift for all of you from Spain. It is a bracelet of our association with the hopeful phrase, lo imposible solo cuesta más. It means the impossible only costs more. And there is another with the phrase, una mirada al futuro, a look to the future, for the research. We hope you like it, and thanks again all of you.
Thanks so much. We hope you'll join us on the terrace.